This is DNA Technology and Genomics, Chapter 20 in the AP Biology textbook. Manipulating genes. We have to talk about recombinant DNA. That means DNA that has come from two different sources and they're combined or recombined to make something new. It's utilized in genetic engineering and biotechnology. So we're going to look to the bacteria model system for this. And we look at simple circular DNA. Simple circular DNA can multiply quickly. It will pick up and keep, the bacteria themselves will pick up and keep what is called a plasmid, which is a small foreign circular DNA, if it's helpful. An example of helpful would be antibiotic resistance. And scientists have noticed that bacteria do this, and they decided to take advantage of it, if you will. So a gene that confers antibiotic resistance. And we've worked with that when we did our PGAL and PGLOW labs, looking at the operons. We used ampicillin resistance as a way to select the bacteria that had our gene. So in this diagram, you have the bacteria with the bacterial DNA in red and some plasmids in blue circular plasmids. Notice they are indeed smaller. Now let's talk about restriction enzymes. Restriction enzymes are used by bacteria to cut out foreign DNA. So if some foreign DNA does get in, they do have a way, an arsenal, an immune system, if you will, to get rid of it if they don't like it. And it will always cut at specific sites, and they're called, they're usually palindromes, and a palindrome is something like this, GAATTC. If you look at these other strand, it would read the same but backwards, and that's a palindrome. It's a mirror image. And so let's look at the EcoR1 restriction site. EcoR1 cuts between the A's and GAATTC. And you can cut it so that you can insert a gene of interest that you want to make copies of. And you can put it into this plasmid DNA. And there's something called sticky ends. They're overhangs, so it doesn't cut directly through like a straight line. It's a jagged edge. And this will allow you to control how that DNA is inserted into the plasmid. So here's a diagram to give you a better idea. So this GAATTC is cut with the EcoR1, and you notice that we have an overhang with the A's and the T's, and this allows us to insert another piece of DNA in the gray here, that, for example, that it does have the similar ends so that they will fit together, and at the bottom we have our recombined DNA that's got a little bit of our plasmid DNA and a little bit of our DNA of interest. Well, now we can look at something called a RIFLEP. A RIFLEP stands for Restriction Length Fragment Polymorphism. That's a mouthful. Well, what does that mean? Well, restriction enzymes cut DNA into different length pieces. So they'll cut wherever there's a palindrome, and in between those palindromes are different length segments of DNA. So if you look at this diagram, if you cut parents and their offspring, you'll see that they'll have inherited half of each parent's, not only their genes, but their restriction fragments, which are located in the non-coding DNA. RIFLEPs are actually made of VNTRs, which are within the non-coding DNA, and that stands for a variable number of tandem repeats, and they're nonsense sequences. And so if you look at that, what that means, variable number means that some people will have maybe 10, another person might have 20 of these repeating sequences that don't code for anything. They're tandem, so they're in line like a tandem bicycle. An example of a VNTR would be AATTAG. One person might have, like I said, 10 of these within their fragment, and another person might have 20. You can compare these, the RIFLEPs of individuals, in a DNA fingerprint. No two people are identical except for identical twins. And so this is why the DNA fingerprinting has become so important in court cases, because they're very, very, very specific. And we do this by a process called gel electrophoresis. We will be performing restriction analysis, DNA fingerprint by gel electrophoresis after vacation. And so here's a diagram to show you. You load your samples in the wells, which are at one end of the gel, and then you apply electricity, and it will cause the DNA to separate, and it'll go through this agarose, which is it's a seaweed, like a gelatin, and they will separate based on their size. And so if you look at figure 2, you'll notice that there's a bunch of different pieces, different size pieces. The larger pieces do not move as far during the same amount of time, and they will all move from the negative end to the positive end, because the DNA is negatively charged and it is attracted to the positive end. And then we can look at it with the UV light. If we have you have to add a little something to it so that it's visualized under UV light. You can also stain it. We're going to probably use the UV light because we can see it in the same day. This is a sample DNA fingerprint. I made it kind of easy for you to look at. Mom is in blue, and you can see that she has a number of different fragments. And Dad is the yellow, and he has fragments that are actually different from Mom's. They're different sizes. And if you look at daughter 1, she has half of her fragments come from Dad and half from Mom, as you would expect. Same with son 1. But if you look at daughter 2, there's a funny thing going on. She has half of hers match with Mom, and then the other half don't match with Dad. You be the judge. What do you think happened? And same with son 2. He doesn't match with anybody. What do you think is going on with him? Southern hybridization. When you run your samples on the gel, it's really hard to see them because you get so many bands if you think of all the possible DNA. So you transfer the DNA to a special kind of paper. It's called nitrocellulose. And then you incubate it with a radioactive probe. You can also use a fluorescent probe. And this probe base pairs with a specific sequence that you're looking for. And then you can expose this to X-ray film, and then you can see only where the probe stuck. So then you don't have to look at millions of bands. It's easier to visualize what you're really seeing. So this is a diagram from your book of first you run your gel. They put it under the biology book. We actually would do this, use liquid to pull it onto the paper, and you have a lot of padding, absorbent paper to pull the liquid, wick it through. And then you put it in the bag with the radioactive probe, and then it shows that step 5 that you can see the bands that were radioactive. PCR stands for polymerase chain reaction. This is DNA replication in a test tube. It was the guy who came up with it, his name is Kary Mullis, and he won a Nobel Prize for thinking of this idea. And this diagram here shows that you split your double-stranded DNA, and then you add your primer, and then you make copies. You add by adding the bases, and then you repeat. So here's the steps. First, you separate the strands. To do that, you have to heat it to 90 degrees Celsius to separate the hydrogen bonds. Then you anneal at 50 degrees. This is approximate, and it varies depending on the primer, but this allows the primer to bind specifically. And then you lengthen or add all your bases at 72 degrees Celsius. And you repeat this for 25 to 30 cycles so that you get enough DNA. PCR is super important because if you think about finding an eyelash and they can figure out whose eyelash it was, there's not enough DNA in that eyelash. You actually need to make lots of copies of it so that you can analyze it. So PCR has revolutionized our understanding of the world.